The brain is an organ that serves as the center of the nervous system in all vertebrate and most invertebrate animals. It consists of nervous tissue and is typically located in the head, cephalization, usually near organs for special senses such as vision, hearing, and olfaction. Being the most specialized organ, it is responsible for receiving information from the sensory nervous system, processing that information, thought, cognition, and intelligence, and the coordination of motor control, muscle activity and endocrine system. While invertebrate brains arise from paired segmental ganglia, each of which is only responsible for the respective body segment of the ventral nerve cord, vertebrate brains develop axially from the midline dorsal nerve cord as a vesicular enlargement at the rostral end of the neural tube, with centralized control over all body segments. All vertebrate brains can be embryonically divided into three parts: the forebrain, prosencephalon, subdivided into telencephalon and diencephalon, midbrain, mesencephalon, and hindbrain, rhombencephalon, subdivided into metencephalon and myelencephalon. The spinal cord, which directly interacts with somatic functions below the head, can be considered a caudal extension of the myelencephalon enclosed inside the vertebral column. Together, the brain and spinal cord constitute the central nervous system in all vertebrates. In humans, the cerebral cortex contains approximately 14 to 16 billion neurons, and the estimated number of neurons in the cerebellum is 55 to 70 billion. Each neuron is connected by synapses to several thousand other neurons, typically communicating with one another via cytoplasmic processes known as dendrites and axons. Axons are usually myelinated and carry trains of rapid microelectric signal pulses called action potentials to target specific recipient cells in other areas of the brain or distant parts of the body. The prefrontal cortex, which controls executive functions, is particularly well developed in humans. Physiologically, brains exert centralized control over a body's other organs. They act on the rest of the body both by generating patterns of muscle activity and by driving the secretion of chemicals called hormones. This centralized control allows rapid and coordinated responses to changes in the environment. Some basic types of responsiveness such as reflexes can be mediated by the spinal cord or peripheral ganglia, but sophisticated purposeful control of behavior based on complex sensory input requires the information integrating capabilities of a centralized brain. The operations of individual brain cells are now understood in considerable detail, but the way they cooperate in ensembles of millions is yet to be solved. Recent models in modern neuroscience treat the brain as a biological computer, very different in mechanism from a digital computer, but similar in the sense that it acquires information from the surrounding world, stores it, and processes it in a variety of ways. This article compares the properties of brains across the entire range of animal species, with the greatest attention to vertebrates. It deals with the human brain insofar as it shares the properties of other brains. The ways in which the human brain differs from other brains are covered in the human brain article. Several topics that might be covered here are instead covered there because much more can be said about them in a human context. The most important that are covered in the human brain article are brain disease and the effects of brain damage. Function. Information from the sense organs is collected in the brain. There it is used to determine what actions the organism is to take. The brain processes the raw data to extract information about the structure of the environment. Next, it combines the processed information with information about the current needs of the animal and with memory of past circumstances. Finally, on the basis of the results, it generates motor response patterns. These signal processing tasks require intricate interplay between a variety of functional subsystems. The function of the brain is to provide coherent control over the actions of an animal. A centralized brain allows groups of muscles to be co-activated in complex patterns. It also allows stimuli impinging on one part of the body to evoke responses in other parts, and it can prevent different parts of the body from acting at cross purposes to each other. Perception. The human brain is provided with information about light, sound, the chemical composition of the atmosphere, temperature, the position of the body in space, proprioception, the chemical composition of the bloodstream, and more. In other animals additional senses are present, such as the infrared heat sense of snakes, the magnetic field sense of some birds, or the electric field sense mainly seen in aquatic animals. Each sensory system begins with specialized receptor cells such as photoreceptor cells in the retina of the eye, or vibration-sensitive hair cells in the cochlea of the ear. The axons of sensory receptor cells travel into the spinal cord or brain, where they transmit their signals to a first-order sensory nucleus dedicated to one specific sensory modality. This primary sensory nucleus sends information to higher-order sensory areas that are dedicated to the same modality. Eventually, via a way station in the thalamus, the signals are sent to the cerebral cortex, where they are processed to extract the relevant features, and integrated with signals coming from other sensory systems. Motor control. Motor systems are areas of the brain that are involved in initiating body movements, that is, in activating muscles. Except for the muscles that control the eye, which are driven by nuclei in the midbrain, all the voluntary muscles in the body are directly innervated by motor neurons in the spinal cord and hindbrain. Spinal motor neurons are controlled both by neural circuits intrinsic to the spinal cord, and by inputs that descend from the brain. The intrinsic spinal circuits implement many reflex responses, and contain pattern generators for rhythmic movements such as walking or swimming. The descending connections from the brain allow for more sophisticated control. The brain contains several motor areas that project directly to the spinal cord. At the lowest level are motor areas in the medulla and pons, which control stereotyped movements such as walking, breathing, or swallowing. At a higher level are areas in the midbrain, such as the red nucleus, which is responsible for coordinating movements of the arms and legs. At a higher level yet is the primary motor cortex, a strip of tissue located at the posterior edge of the frontal lobe. The primary motor cortex sends projections to the subcortical motor areas, but also sends a massive projection directly to the spinal cord, through the pyramidal tract. This direct corticospinal projection allows for precise voluntary control of the fine details of movements. Other motor-related brain areas exert secondary effects by projecting to the primary motor areas. Among the most important secondary areas are the premotor cortex, supplementary motor area, basal ganglia, and cerebellum. In addition to all of the above, the brain and spinal cord contain extensive circuitry to control the autonomic nervous system, which controls the movement of the smooth muscle of the body. Sleep. Many animals alternate between sleeping and waking in a daily cycle. Arousal and alertness are also modulated on a finer time scale by a network of brain areas. A key component of the sleep system is the suprachiasmatic nucleus, SCN, a tiny part of the hypothalamus located directly above the point at which the optic nerves from the two eyes cross. The SCN contains the body's central biological clock. Neurons there show activity levels that rise and fall with a period of about 24 hours, circadian rhythms. These activity fluctuations are driven by rhythmic changes in expression of a set of clock genes. The SCN continues to keep time even if it is excised from the brain and placed in a dish of warm nutrient solution, but it ordinarily receives input from the optic nerves, through the retinohypothalamic tract, RHT, that allows daily light-dark cycles to calibrate the clock. The SCN projects to a set of areas in the hypothalamus, brainstem, and midbrain that are involved in implementing sleep-wake cycles. An important component of the system is the reticular formation, a group of neuron clusters scattered diffusely through the core of the lower brain. Reticular neurons send signals to the thalamus, which in turn sends activity level controlling signals to every part of the cortex. Damage to the reticular formation can produce a permanent state of coma. Sleep involves great changes in brain activity. Until the 1950s it was generally believed that the brain essentially shuts off during sleep, but this is now known to be far from true. Activity continues, but patterns become very different. There are two types of sleep: REM sleep, with dreaming, and NREM, non-REM, usually without dreaming, sleep, which repeat in slightly varying patterns throughout a sleep episode. Three broad types of distinct brain activity patterns can be measured: REM, light NREM and deep NREM. During deep NREM sleep, also called slow-wave sleep, activity in the cortex takes the form of large synchronized waves, whereas in the waking state it is noisy and desynchronized. Levels of the neurotransmitters norepinephrine and serotonin drop during slow-wave sleep, and fall almost to zero during REM sleep. Levels of acetylcholine show the reverse pattern. Homeostasis. For any animal, survival requires maintaining a variety of parameters of bodily state within a limited range of variation. These include temperature, water content, salt concentration in the bloodstream, blood glucose levels, blood oxygen level, and others. The ability of an animal to regulate the internal environment of its body, the milieu intérieur, as the pioneering physiologist Claude Bernard called it, is known as homeostasis, Greek for standing still. Maintaining homeostasis is a crucial function of the brain. The basic principle that underlies homeostasis is negative feedback. Any time a parameter diverges from its set point, sensors generate an error signal that evokes a response that causes the parameter to shift back toward its optimum value. This principle is widely used in engineering, for example in the control of temperature using a thermostat. In vertebrates, the part of the brain that plays the greatest role is the hypothalamus, a small region at the base of the forebrain whose size does not reflect its complexity or the importance of its function. The hypothalamus is a collection of small nuclei, most of which are involved in basic biological functions. Some of these functions relate to arousal or to social interactions such as sexuality, aggression, or maternal behaviors, but many of them relate to homeostasis. Several hypothalamic nuclei receive input from sensors located in the lining of blood vessels, conveying information about temperature, sodium level, glucose level, blood oxygen level, and other parameters. These hypothalamic nuclei send output signals to motor areas that can generate actions to rectify deficiencies. Some of the outputs also go to the pituitary gland, a tiny gland attached to the brain directly underneath the hypothalamus. The pituitary gland secretes hormones into the bloodstream, where they circulate throughout the body and induce changes in cellular activity. Motivation. The individual animals need to express survival-promoting behaviors, such as seeking food, water, shelter, and a mate. The motivational system in the brain monitors the current state of satisfaction of these goals, and activates behaviors to meet any needs that arise. The motivational system works largely by a reward-punishment mechanism. When a particular behavior is followed by favorable consequences, the reward mechanism in the brain is activated, which induces structural changes inside the brain that cause the same behavior to be repeated later, whenever a similar situation arises. Conversely, when a behavior is followed by unfavorable consequences, the brain's punishment mechanism is activated, inducing structural changes that cause the behavior to be suppressed when similar situations arise in the future. Most organisms studied to date use a reward-punishment mechanism. For instance, worms and insects can alter their behavior to seek food sources or to avoid dangers. In vertebrates, the reward-punishment system is implemented by a specific set of brain structures, at the heart of which lie the basal ganglia, a set of interconnected areas at the base of the forebrain. The basal ganglia are the central site at which decisions are made. The basal ganglia exert a sustained inhibitory control over most of the motor systems in the brain. When this inhibition is released, a motor system is permitted to execute the action it is programmed to carry out. Rewards and punishments function by altering the relationship between the inputs that the basal ganglia receive and the decision signals that are emitted. The reward mechanism is better understood than the punishment mechanism, because its role in drug abuse has caused it to be studied very intensively. Research has shown that the neurotransmitter dopamine plays a central role. Addictive drugs such as cocaine, amphetamine, and nicotine either cause dopamine levels to rise or cause the effects of dopamine inside the brain to be enhanced. Learning and memory. Almost all animals are capable of modifying their behavior as a result of experience, even the most primitive types of worms. Because behavior is driven by brain activity, changes in behavior must somehow correspond to changes inside the brain. Already in the late 19th century, theorists like Santiago Ramón y Cajal argued that the most plausible explanation is that learning and memory are expressed as changes in the synaptic connections between neurons. Until 1970, however, experimental evidence to support the synaptic plasticity hypothesis was lacking. In 1971, Tim Bliss and Terje Lomo published a paper on a phenomenon now called long-term potentiation. The paper showed clear evidence of activity-induced synaptic changes that lasted for at least several days. Since then, technical advances have made these sorts of experiments much easier to carry out, and thousands of studies have been made that have clarified the mechanism of synaptic change, and uncovered other types of activity-driven synaptic change in a variety of brain areas, including the cerebral cortex, hippocampus, basal ganglia, and cerebellum. Brain-derived neurotrophic factor, BDNF, and physical activity appear to play a beneficial role in the process. Neuroscientists currently distinguish several types of learning and memory that are implemented by the brain in distinct ways. Working memory is the ability of the brain to maintain a temporary representation of information about the task that an animal is currently engaged in. This sort of dynamic memory is thought to be mediated by the formation of cell assemblies, groups of activated neurons that maintain their activity by constantly stimulating one another. Episodic memory is the ability to remember the details of specific events. This sort of memory can last for a lifetime. Much evidence implicates the hippocampus in playing a crucial role. People with severe damage to the hippocampus sometimes show amnesia, that is, inability to form new long-lasting episodic memories. Semantic memory is the ability to learn facts and relationships. This sort of memory is probably stored largely in the cerebral cortex, mediated by changes in connections between cells that represent specific types of information. Instrumental learning is the ability for rewards and punishments to modify behavior. It is implemented by a network of brain areas centered on the basal ganglia. Motor learning is the ability to refine patterns of body movement by practicing, or more generally by repetition. A number of brain areas are involved, including the premotor cortex, basal ganglia, and especially the cerebellum, which functions as a large memory bank for micro-adjustments of the parameters of movement. Thank you for watching this video.